Okay, genetics and inheritance, paternity testing, guys. Today we'll be doing past examination questions because you did paternity testing back during term 1 when you were doing DNA profiling, right? So welcome to my class, guys. Life Science Grade 12 2024. Today's date 10 April 2024. It's Wednesday, and the time there it's exactly 19 minutes past 12. So welcome, guys, and bonani, dumelani, avsheni, hello, nda, varmini. So okay, guys, for those who are new here, we welcome you. Feel free to subscribe, feel free to like, feel free to comment, guys. And we also have these learners who are engaging, although we are at 45 40 40 something, 46 45 subscribers, guys. We have Bolulu Lululo, Affectionate Chocolate, Arnold Botedi. These are the accounts that are engaging to our group, right? We also have Naledi Makamedi. So if also you want this kind of shout out, you can comment on our comment section, right? So we also have this question here. Someone asked me, is there any WhatsApp or Facebook group for subscribers? So for this message, guys, today make sure that you send me a 0711906168. So let's do like this. You send me a screenshot subscription via WhatsApp a private account like this, and then now I will add you straight to the WhatsApp group. Agreed? Okay, you just send a subscription, and after subscribe, after the that thing that shows that you subscribed there, you just screenshot that part with my African transcribers on top, and then you send it to me via screenshot, just that, and then that will be your invitation to our group. So with that being said, thank you, guys. Welcome to my class. Those who have been subscribed, hit that subscription button and hit that notification button, so every day after I submit my videos you'll be notified. So for sure, every day I'll post them. There will be around around 5, around 6 available for you guys when you come back from school, right? So thank you. Week 4, today we are doing week 4, right? Paternity testing. That's an yesterday, guys. I would like to apologize for yesterday, guys. Yesterday I did close to 2 hours video. If you missed it, go and watch it, guys. We had a full almost 2 hours of non-stop genetic engineering and stem cell, right? And then I'd like to apologize for that. This time around I'll have to keep my track of time and plan my things accordingly. Likely we are still on our track as we promised, from Monday, Tuesday, Wednesday, Thursday, Friday. That Monday we'll do genetic engineering, Tuesday we'll do stem cell cloning, here we're doing a recombinant DNA, and then Wednesday we are going to do paternity testing, but including it on our question papers, right? Question and answer papers, because you know that Wednesday we have this session here, and then Thursday we are going to do a mitochondrial link, and then this will mark our end of a genetic link, which is mitochondrial links, right? So this will mark the end of this week, and then we'll do some revision, and then after that Friday we'll come and do the activity. We planned this, we were even talking about this previously. So if you are coming here after viewing my video, guys, I appreciate you, and I really like to promise you that I will dedicate my time to research and maybe understand this life science better, so that I will also come here and explain to you in an understandable manner. So with that being said, guys, kupulole subscribe being there, and then paternity testing. You did paternity testing test 1, term 1, right? And then in term 2 also, it's been included here. It means that even this term 2, we are still going to ask you questions related to paternity testing. DNA, you know that we can use a DNA to DNA profile to check or to compare the relativity between two individuals, right? So with that being said, with our caps we are done this week. Paternity testing and genetic fingerprinting here, the last part here. Then we do is that this one, it's used as a forensic mostly. So we also use this for different things, right? You agree with me that we also use a paternity testing or DNA but fingerprinting, yeah, DNA filing, yeah, same thing, right? Proofing, DNA proofing. profiles. Right, thank you. And then as you can see with our examination guideline, let's take it with our examination guideline, you did this year, term 1, right? DNA profiling. And then you have to learn to interpret the DNA profile and uses of DNA profiles, right? So here you can name them, that we use these profiles for tracing missing person, we use this to identify genetic disorders, we use this to identify dead people, you know, by this time, right? So that's the information that you'll have to take from term 1 and then you bring it here in term 2 under week 4, right? So you can see with our examination guideline that we are done with we are almost done with our term 2 week 4, right? First topic. And then we are going to do paternity testing, but the use of each of the following paternity testing: blood grouping, right? Yesterday I did give a small brief blood grouping. We did blood grouping. We also have a full video under blood grouping, right? So if you missed blood grouping, go back there, and then you can also learn how we use this blood grouping to identify a paternity right, paternity relationship between a parent and the offspring, right? So an example, I did an example here. If, like, for an example, your father is having an A and B, and then your mother, she will be having, or she was having A and B, and then now you are blood group O, right? So with this information, you can conclude that you were adopted in this family, right? So a simple thing like this, we can use this to identify whether maybe I was adopted or I'm the real genetically you understand, offspring of my parents. So you have to learn that, and you have to understand that. We also have to remind you here, term 2, right? And then another thing, it's blood DNA profile. Since we did blood grouping, I'll say this again, since we did blood grouping, today we are going to leave blood grouping behind and we are going to do DNA profile, since we didn't do DNA profile. The same question will come, the same thing that I'm asking here is the same thing that we did under blood grouping, and it's the same thing that they are going to ask you. If need be, they ask you question related to blood grouping. So DNA profile, let's go. Yesterday, Monday and Tuesday, we did all this that you see here, crashed. We did this biotechnology and genetic engineering, stem cell research, genetically modified organism, and then we did cloning, right? So you will allow me to move on. Even on our green book, if you still remember, we did this activity here. This one here, pedigree, right? Let me remind you, we did this pedigree activity. Then after doing this pedigree activity, after that, our green book just said 5.5 genetic engineering, and then they gave you a brief explanation of what is genetic engineering. Genetic engineering is the process whereby genes on the DNA are changed, right? Manipulated and then transferred. Yes, you can also, or manipulated to produce a different organism, right? This is genetic engineering. We are manipulating a gene to produce a different organism or to produce a byproduct of a biological processes, right? And then after that, it gives you the advantages and disadvantages of this genetic engineering. Why? Disadvantage here, they said it's expensive. We talked about this, right? Expensive, you got it right there. Then from there, the potential of health impact, interfering with nature. You see immoral here, those people who are so religiously, they will interfere with this, or they will go against this, saying that this is immoral, you understand? And then we are interfering with nature. And then there is unsure of a long-term effect, right? I even told you that you might find that you are introducing a new gene to our DNA, and then later on, the later stage, we have an effect somewhere, right? And then advantages, they said they can produce medication, we were right. And then they can control pests, and then using specific genes to increase crop yield, right? Or food security. This can be crop yield, yields. When it increases, it means that we have more food, or whatever we are producing, we have more, right? And then you can selecting genes to increase shelf life of a plant. So these products that are produced under genetic modification, they have a long shelf life, right? So one, they are not easily to be affected by diseases. Two, they are not easily to be You understand? Those we did yesterday, right? And then here we have another one that this one, yeah, they're not easily to get spoils. It means that we increase our shelf life. Shelf life, it means that inside the shops where the food will be placed, they will stay longest time without rotting, right? This is the shelf life. Shelf yona ila ya lebenkele. So you'll agree with me that we did all this, right? And then from there, they told us that you can be a genetic counselor, and then they gave you a small scenario of a couple that they undergo genetic counseling to enable them to make informed a decision on whether they want to have children or not, right? So when now you'll know that with this information, you can use the information that you'll get here they say to give advices. You can get advices. They can ask you the advantages or benefits. Yeah, state 3 benefits genetic counseling in this form, right? So this will be, you will given an explanation, you'll given an option, and then they will make you to understand whether to have a baby. You understand? For an example, if I'm saying that now I'm having tall legs, for an example, I'm having tall legs and my wife, she's having short legs, and then we can go to the DNA counselor or genetic counselor, and then we inquire or we consult whether we can have babies that are having the long legs or the short leg. So with our knowledge of DNA and genetic inheritance, we can now study where this long feet is coming from, right? Pedigree and what, and then this side we can also study that layer, and then we see which one is dominant, which one is recessive. If this one is dominant, we can now give the informed decision to those couple that there's a high possibility of your children or your offspring to have long legs, right? We are just passing by here, right? Hence you can see that I'm speeding like what. So then after that, we'll be moving to response to the environment by humans next week, right? So here in this chapter we are done. You see, they didn't even include a sperm cell, and they didn't even include cloning and the genetic DNA, recombinant DNA, right? So likely because you have me, we also did this here under our textbook. We did biotechnology. Today we'll do a DNA pro. You can see even on our textbook, they just include DNA profiling, right? And then they said you did this in chapter 1, and then previously we were doing blood grouping, so now we'll do DNA, so that we'll cover everything accordingly. Then biotechnology, we did biotechnology, the use of organism, right? Then from there we did genetic engineering. We talked about this recombinant DNA. We did recombinant DNA advantages and disadvantages. We did this stem cell technology. We are just passing by, right? And then I even gave you an activity, this one, activity 8. If you haven't done this, guys, I will give you also today, so that you can also do this. We'll come and do this tomorrow before we do the mitochondrial links, right? Before we do this last part here, right? So with that being said, guys, thank you for being here. Whew. Now we can move to our previous examination, right? So life science, this question paper was written in 2021 November, life science paper 2, right? So I collected some questions so that we can do them together as today's class, right? So I think we are now doing these past examination papers, right? Preparing and reminding you of DNA proofing, how it was supposed to be done. If they taught you, we are fine. If you are lacking, you are here. Don't forget to subscribe, guys. Don't forget to follow me on my TikTok. Don't forget to follow me on my WhatsApp and my Facebook, right? And then 1.1.5, this is where we'll start with blood groupings. So we'll start with blood groupings. After blood groupings, we can now go to how these activities, genetic engineering and stem cell, right? They use those questions mostly on investigation, right? They'll give you a small investigation, and then now they ask you the question. I'll show you. I'll do both questions here, right? I'll do both questions, and then we can see. And then those recombinant DNA, same as like that, right? When they ask you about a briefly outline the recombinant DNA, you said those we were doing yesterday. You missed that, go back, guys. We no longer have time. I spent lots of time yesterday covering those two topics, right? So here's our activity today. And then 1.1.5, Okay, but in a car accident, right, the biological father of a child and three other men were killed. You still remember that previously, I'm not sure if it was Monday or Tuesday, I did an example of this, right, using that example of that bus that got accident there going to Moria. You still remember that if we were to say we are going to identify those people, how were we going, how you're not you, that is the English, how we were going to find out uri monkey man wako kai, who's who from where, right? So the same question, I found the same question here, 2021. The question was asked again. It was asked, right? It was not again because this was before me coming with it, but I found it here. But in a car accident, the biological father of a child and three other men were killed. It means that there were four, right? All of them, they died, right? So the men could not be identified due to their injuries. Maybe the car burned, right? Something like that. We can think of that. Sorry, but we can, you understand? Even this, we can also relate to what was happening previously. Rest in peace, and to their families, our condolences. And then here's the thing. But the men could not be identified due to their injuries. But DNA profiling was used to identify the girl's father, right, amongst this. So we are not sure if the girl was the driver. We can say this one man 1, and then man 2, man 3, man 4. So here we are not sure whether man 1, maybe they got crashed or something, right? So we are not sure. Hey, I'm starting to think many scenarios. Still remember the one that the truck bombed there and burned people, even the inspector. But yeah, so not say distant buys. Still remember that incident. So yeah, this is what we can use to identify those people. So we can say there's number 1, number 2, number 3, number 4. We are not sure who is who, right? So they want to see, or this girl, they want to identify amongst these 4 individuals which one was this girl's father, right? And then the diagram below shows the DNA profiles of the girl, their mother, and the 4 men. So here's what they happen. Here's what's happened, right? I even told you that if we were going to identify each and every person, the DNA of that bus, we are going to collect all 46. If there were 60, I'm not sure how many were there, right? If there were 43, 43 of them, we are going to collect all 43 samples, right? And then we name them sample number 1, number 2, number 3, number 4, number 5, up until 43. And then here we sampled the DNA of the girl, and we sampled the DNA of the mother. Life science word. Sample is to take. When you say you go to a cell and you take a certain gene, you are sampling, right? To sample, they will say, hey, they sample. It means that they take aside that specific thing. Just they take. Yes, we took a girl DNA and the mother DNA. As you can see, firstly, you learn this, right? You learn this. We even learn that this it also, even though it's 100% way to test paternity testing, this can also come with errors because we use human observation skills to observe and interpret this. So you understand, with human we can make mistakes. So let's interpret this. We have as you can see, we have a mother, we have a girl and a mother. Yes, we'll start, right? And then we have 4 men this side. And then if you can see the girl here, the girl, she have the band here, and then the girl, she also have a band. These are called the bands, right? We call them the bands. This is a revision yana. This we call them the bands, right? Or the bar. You can just call them the bands or the bar, right? And then we also see the girl here, a bar number 3. And then we also hear the girl, bar number 4, bar number 5, right? Bar number 1, bar number 2, bar number 3, bar number 4, bar number 5, right? So we also sampled the mother's DNA. Why are we sampling the mother's DNA? Because we only want the father's DNA and the girl DNA. This mother's DNA is to remove the bars here that the girl inherited from the mother. So from this, we are going to say this sample number, this band number 1, we can conclude that she inherited it from the mother, right? Then from there we see this one is not there and this one is there from the mother and this one from the mother. So if it's like this, let's go and check those that she might have inherited from the father, right? So we go to this band here, band number 2. We go to band number 2, we go with this line matching band number 2, we find that we have a band number 1 right there. Then we write number 1. This is not the final answer. We still have to find another band, right? And then we go again with this band. We can say this band there's a possibility of this girl to inherit this band from man 1, or she can inherit it from man 2, right? So now we can conclude maybe man 1 and man 2, they were relative in this car accident, right? These are the other information in number 2. These are the other information that we can come with, right? And then we go with another band that there's a possibility of her to only get it from the father. This band number 3, we go straight with it, and we see that number 2 there is that band there, and then number 4 there, number 3 there, we also have that band here. Number 3, number 4, we also have this band here, right? And then we go to the last band. This one, she inherited from the mother. We expect it not to find it from the father and the father. And by number 5, there's a possibility of her to inherit it from the mother, and there's a possibility of that band to be inherited from the father here. This conclude that man number 3 there, he's our father to this girl, right, using our interpretation. And then what with these two we can conclude that maybe they are the relevant to the main 3 here, because you can also see that this band here, we also find it here, right? So there's a possibility, if we are saying number 2 there's a possibility of number 2 to be the father here. We are going to say she inherited this from the father but not the mother, right? So it means that even this one we are going to crash it. Then we're left with this 2 number 4 being our decider of which one is which. And then but which one of the men is most likely to have been the father of the girl. And then number 3 is number B, right? So that was it. So this is how we interpret this DNA profile, right? What do we use this DNA profile for? We have different uses of this profile, right? So thank you. Let's move on and get other questions that they might be dealing with this DNA profiling, right? We don't have them. Go to our next page. We have it here, right? 2.5, and then this page is 2022. This question paper 2022. Yes, the question paper, Life Sciences paper 2, November 2022, right? So let's go to our DNA profiling and see how we were going to interpret. We also have 2.3 there. We'll come back, right? We'll come back because that one is related to genetic and what. So let's start with this first. Then 2.5 there. In the 2.5, it says the diagram below represent the DNA profile of 3 children, right? And their parents. So here we have the parents, and we have 3 children, right? And then only 2 of the children, here's the statement again, only two of the children are their biological children, and the one is adopted. Okay. It means that we have the father and we have the mother. Sorry, we have the mother here. And between Haila, these 3, Haila, Priya, and Leo, one of them, the baby, she is adopted. If both of them they are if both of them they are females, right? So we'll take them as we'll understand also which one is a boy, which one is a. Moving on, but we can just conclude all of them they are female, Haila, Priya, because they didn't tell us. Either way, that information is not necessary under these questions. And then 2.5.1, Barry, identify the 2 biological children, right? Let's identify the 2 biological children. Which children are these? Between these 3 of them, which of these 2 they are the biological children, right? So let's exclude the one that we think this person, he or she, is an adopted. So let's go to our band. As we can come here with our mother, we know that we have the first band here. We go with it, this band, we go this band. We find that, oh, also Haila, she also have this band, right? So we can now come and say 1 here. But this does not conclude that Haila, she is the biological mother. You might find that this is a band maybe carrying a DNA for a female reproductive what. You understand? So you know that in somatic cell we are going to collect all 46 chromosomes, and this is 46 chromosome, we have X and X there. So what if this band, it's that DNA there at X and X, that only stipulate that Haila, she's a female. So this is not our final conclusion. So we can go again and say father here, the father here is also having a band here, and this band we can also find it in Heila. These two now lock us to say our first child there ki Heila. First just there ki Heila. Then let's go now and find another child, right? Now we have to exclude Priya and Leo. Just have to come and exclude these two. Is this band from the mother and son, from the father and the mother? No. Right, you can see it's in between these two, right? Is this band in father or the mother? We can see that yes, this band is available here from the father, right? Let's see another band that is at Leo, that we can find it from the father and the mother. We also have the last band here. The last band here is the same as Hela, and it's also same as Leo, which conclude now 100% that these two, they are siblings and they are the baby to this mother. So Leo, Hela, and Leo, they are there. They are two biological children. Hence, we have 2 marks there. So we can, you can say, they can say identify the adopted child. Bria was going to be the answer. Then 2.5.2, but explain your answer to question 2.5.1. So if now you didn't guess this, if you didn't come with Magezelano, because they can, maybe you can come and say Hela and Priya, right? Or say Priya and Leo. Because you have one of the name, they can say give one answer to one name, right? So if you wrote Priya, I want to give this example. If you wrote Priya and Leo, and then we want one answer, this one will be wrong and Leo will be right, right? So if you got both of them, it means that you studied this. So now we want you to explain your answer here. If you wrote Priya and Leo, you are going to explain your answer here, Priya and Leo. Maybe you might say, hey, I'm not sure how you are going to explain this one here, Priya and Leo, because I can say I can say. So I can say, well, now you're going to find a way to explain this one here, Priya and Leo. But right now we chose Hela and Leo. So we are going to explain here Hela and Leo. We can say with Hela and Leo, they share the last band, right? They share the last band or the last bar. We see the same bar from Leo and Hela and the mother, right? And the mother. Since there's many ways to kill a cat, you can also say in Hela, there's band number 2. It's also traceable from the father, band number 2. And then in Leo, also in Leo, band number 2, we can also trace it back to the father, band number 2. You are killing one cat with, you are killing a cat with many ways, right? Or you can say band number 2, Leo, band number, this one is band number 1, 2, 3, band number 3, yeah, Leo is at the same position as band number 1, 2, 3, yeah, Helia, and both this band we can find them at band number 1, 2, 3, yeah, father. Right, when you get your 2 marks. So you see that there's many ways to explain how you came up with Hela and Leo. So when you explain it in this way, it means that you are heading to the right direction, right? So since we have three from the father and one from the mother, it means that these 3, they are the biological, these 2, they are the biological parents to Hela and Leo. So 2.5.3, state 3 other uses of DNA profiling. State other 3 uses. We can say number 1, paternity, or they say other, right? Other uses. So they won't give you if you do paternity testing, because you were doing paternity testing. Give us other 3. Right? We can trace missing person. Right? We can trace missing person. Example, maybe like we are saying someone was missed, and then that person, for example, we find a baby, and then finding that baby, and then we want to identify their biological father. Since we have many people who go around today reporting the cases of missing people, we'll have to use DNA to identify the real biological parents to that daughter, or else we can just take that daughter and give any other parents, since that family reported that there's a missing daughter. I hope you understand whatever I'm trying to say there. We can do this to trace missing person or to identify the dead, right? To identify the dead. To identify the dead or to identify dead person, right? Whether the person who is dead is related to a certain people or not, right? And then the third one, since they said three, we can use this to identify the genetic disorder, right? To identify, when I say identify, to identify the genetic disorder, the disorder. When I say to identify genetic disorder, let's say that this band here, this band number 2 ya Leo, right? This band number 2 ya Leo, we found it, maybe we are studying HIV, right? We are studying HIV, and then we find that this band is there, and then it's also there at Helia, and we found it from the father. So we can conclude that Leo, she is a HIV positive, and she inherited this HIV positive from the father. So we can use this to identify the genetic disorders, or maybe using hemophilia, those disorders. Let's use the disorders that we are using, hemophilia and what else? Color blindness and those others, right? So we can say with this band, because it's coming from the father, we can conclude that this person Leo here, she inherited this from the father, right? So we can use this to identify, or we can use to identify the crime, to suspect crime, right? We can also use to identify the suspect of the crime, right? Of the crime. For an example, you did a crime, and then maybe you got a cut, or you left your DNA inside the victim's place. We can go and collect that DNA. After collecting that DNA, we go and find a suspect. So we have to have the suspect. If you don't have the suspect, hence some people, they are not going to be arrested, because we don't have the suspect, and we can't go to the entire world and search the DNA profile. Or we can register a DNA profile of someone into the database, where now your ID number comes with your DNA database, right? The moment something happens, we just go and take the DNA and put it inside the computer, and the computer match the DNA from your ID and the DNA of the suspect, and then we arrest you. You see? So these are other opportunities that we can come up as life science students, right? So from there, they said we can use this to identify the suspect of the crime. I was still explaining how we can identify the guy, and then after collecting the DNA, we collect the samples of the suspect, and then we match, right? Maybe this is the suspect number, this one will be the suspect, and this one will be the suspect DNA, right? Then here we'll have a sample, and then we'll have those suspect here. Then we'll see the bands. If we say that Leo, we can see that Leo, she has a DNA that from a suspect. Then we also, maybe another thing, so that our experiment will be reliable, we can also add the DNA of the families there, so that we don't mix the DNA, so that we can sell this for us to identify whether the DNA that we found there, it's not the part of our families, our family members, right? So we'll have to test them also, so that when we get that DNA, we can exclude them from the family members. Let's say we are 3 in the family, and then Tabo, Na, and then my grandmother, and then something happened, and then maybe we are tracing DNA from a hair, a human hair. You know that they have a DNA there, or we are tracing this DNA from something, right? We got it from someone, then we're tracing this DNA. So for this DNA to exclude it from this tree, we can also collect DNA from this tree, and we match it with our DNA. If we find that this DNA is not matching one of these three, it means that this DNA now is our suspect DNA, right? During rape, you leave your semen there, you know that your semen sperm there, and then that sperm have your 46 chromosomes, your 23 chromosomes. We can take that chromosomes, cut them, and then reach here when as a victim, we collect your sperm, we collect your sperm. Yes, you think here we'll use the sperm that we got from the baby, and then here we'll use the girl's DNA, and then we will come and take your DNA here, right, and compare them, right? If we find that the DNA that we see here, it's not the same as the DNA that we see on this girl, it means that this foreign DNA care the second person, and we find that the DNA immediately that we arrest you, right? So With that being said, we can also use this to match a organ transplant, right? During the organ transplant, maybe like we want to transplant, to remove one organism to place it onto another person. We can use DNA profiling to check the match, right? So with that being said, guys, let's go to our 2023 question paper. 2023, this one, 2023 question paper, November, right? Okay, with also DNA profiling. Here's our question here. It was 1.1.6. The DNA profile, these are the type of high value question, because you'll see, they will need you to come and focus. It's not a guess here. You don't just come and say D because you feel like it's D. You have to come and concentrate and put your whole memories and what engage to your oblongata, engage to your cerebellum, engage to your your cortex and what. We are going to study this next week, guys, when we are doing a response to the environment. We'll be talking about the brain, where everything now is being. The moment you see this question paper, it's sent to your eyes, and then your eyes is capturing this. After capturing this, there's some guys inside your head. They take this thing and then they send it to the brain. After sending to the brain, there's someone in the brain coming and taking this thing and put it down under the computer, and they analyze this thing, and then they tell you that whatever you are looking at now is the whiteboard written. The DNA profile of 4 individuals are given below. WXYZ. That's your brain, my guy. We'll be learning that next week. Hang on. Tshwarelela, my guy. Let's move on, right? Today we are doing DNA profiling. 1.1.6. But the DNA profile of 4 individuals are given below. We have W, we have X, we have Y, we have Z, right? So for sure, we will have to see those that are aligning what in this for what. Then here we can see we have the bar that is aligning into 3 of them, right? This one, it can be first, and then we also have this one. We have the bars that are aligning here. That can be our second clue before we can go and answer our question, right? Then, but which individual are possible members of the family? Didn't we we also have to exclude which individuals are possible members of the family. Here in this group here, we have this, we have this, we have this, we have this, right? So 4 of them. So we also have this, we also have this, we also have this, we also have this, right? So 4 of them. So now let's go and exclude others now, right? These 4, they are just showing that this all of this is the same species, right? That's what we can take here. We can take maybe all of them, they are girls, all of them, they are boys, all of them, they are human beings as an individual. So we want to see those who are related. Then we can see that Y and Z here, they are giving more related, and the number WX here, W, Y, and Z, they have this, that number X, it doesn't have, or she doesn't have, right? So we can conclude now that X, Y, Z, they are the relatives. And also we can also conclude with this bar here, and this bar here, and this bar here. You can see that here she have a different kind of DNA there, right? So more evidence they are excluding number X. So we are going to have a W, Y, and Z. W, Y, and Z. So the answer will be number C. It's only 1 mark, but you see how we have to get that 1 mark, right? So from there, let me check under 2023 if we also have another question related to blood what, right? So we don't have, right? So we have these questions here. I also love to do this, still have time. 2.3, right? These are the questions that under this genetic what, since we are now doing question papers under this engineering here, under this genetic engineering, we will be using, we will be doing the practicals, right? You will be doing practicals, asking you to interpret a graph. So you know that we have the graph, we have the line graph, we have the bar graph, we have the histogram, we have the pie chart, you understand? So you should be able to interpret this, and then you know that when we see this, it's mostly asked under experiment questions, right? They will be asking you the percentage, how many of what was what. So here maybe for example here we can give you we can say date, and then here we can say YouTube video length, right? Video length, right? So you can say Monday, you see that our length, maybe we started with 10 minutes, and then Tuesday we go here, Wednesday, and then today we are here. Now we can cover 2 hours, right? And so in our line graph they can ask you the possibility of this, give us the outline or give us this digit here. Do you understand? So they give us the dependent and independent variables. You get them here, right? The dependent, the one that is depending on something. Independent is the one that is standing alone. Like here we can say a date here independent because it's staying alone, right? It's not depending on something, but the length of our video, they are depending on the date. When the date increase, our video content increases. We can say like that, right? So in this question they'll also ask this, or they can also give you a small picture of a cell. They show a cell here, and then they show a cell here. They show at the second step removing the nucleus, and then even this side removing the only the cover, some this, and then we add these two, and then we have one like this. And then they can ask you what is process that is taking place here. You know, when we take one cell from one cell and then we inject the nucleus here, this process is called cloning, right? Or they can also show you another one where we take a cell, we remove a genome, and then we cut it like this, and then here we remove a gene, we cut it like this, and then we combine this, and then this one is called a recombinant DNA. You still remember, right? So those are the questions that they are going to ask you. These are the manner that they are going to ask this, right? So if I find another questions, I will come and show you what I was talking about, right? But we can also go to our previous question papers, the one that we are doing recently, and see how the questions were asked when they were relating to genetic engineering, right? So with that being said, let's come to 20, this one, right? 2.3. 2022 November, right? 2022 November, we scanned this, we did that, we did this, and then now we are going to do this question 2.3, right? Wait for me, let's see. ASCII 2.3, right? 2.3 are the number of chromosomes in the somatic cells of organism differs from species to species, right? The number of chromosomes, we have learned up to so far that in a somatic cell we have full set of 46 chromosomes in a human being, but they're saying that we know that this number differs from species to species, right? So it means we are 45, maybe the cat is going to have 40, maybe like so, right? A elephant is going to have another number. So the graph below shows the number of chromosomes in each somatic cell, right, of the 3 different species. We have the giraffe, we have the mouse, and then we have the pineapple plant, and then we have the species, right? Species, we can have animal and a plant, both are the species. And the number of chromosomes here, we can see that the number of chromosomes is controlled by the species, right? The number of chromosomes is depending on the species. The giraffe, it's having 30, and then mouse there they are having 40, and the pineapple plant there it's having 50 there. And the number, the head there, you also know that with this you also have to include the heading. If they say maybe represent this answer in a table or in a graph form, first thing you have to have your topping, your heading there, and then from the heading you have to have your y and x axis, right, there placed, right? And then after there you'll have to have your scale. Make sure that your scale there stamina sharp, don't have 10 and then 15, then you understand. Then this will be your answer if you have to represent, right? And then the heading there, but the number of chromosomes in the somatic cells of 3 different species. Where do we get this heading, Mr. Mpepu? If maybe they say draw a bar graph of what, you get it from the experiment here. This is the heading, the number of chromosomes in a somatic cell and organisms, right? You can say the number of chromosomes in the somatic cell of different species. This of 3 different species, right? So we can move on from there. That was a tip nyana. So we can move on from there. And then 2.3.1, but how many chromosomes? And then remember, this one is a somatic cell, right? We have two types of cells. We have somatic cells and we have a gametes, right? We have gametes, both sperm and the ovum, right? So okay, but how many chromosomes will be present in number A, mouse cells during telophase number 2 of meiosis? It means that we say ipmat, and then interphase, first step, the prophase, metaphase, anaphase, telophase. The telophase, the last step, and then they are saying number 2. It means that this will be the final step of meiosis. What do we know about meiosis? We know that in meiosis, if we have 46 chromosomes, they are going to be halved to 23. So using this formula, we go to and halved any chromosomes after telophase 2. Then the number of mouse cell during a telophase number 2, and then mouse here in early 40, right? You can agree with me. So this one is the somatic cells. After undergoing meiosis, we are going to have this 40, and then we are going to have a 20 there, right? Sure, we are going to have 20 there. And then number 2, but a leaf cell, how many chromosomes will be present in a leaf cell of a pineapple plant? A leaf cell of is this a gamete? Now, a leaf cell, it's a leaf cell, a gamete? Answer, it's no. So if it's not a gamete, it's a somatic cell. So they want the number of chromosomes in a leaf cell of a plant. To come here, we said it's 50, then the answer will be 50. You'll agree with me, right? So if they wanted the gametes, like here they were wanting the gametes one, then we know that it will be halved, right? So we're moving on to 2.3.2. But explain why the sperm cell of a giraffe has 15 chromosomes. Where's the giraffe? There's the giraffe. Then we have number 20 in the somatic cell. It will undergo meiosis, and then we have 15. But explain why the sperm cell of a giraffe has a 15 chromosome. Because the sperm cell under the somatic cells, for sperm cell to be produced, or a sperm cell is a byproduct of meiosis. Therefore, a somatic cell 30 somatic cell will divide into 2, and we are going to have 15 gamete cells, 15 chromosomes found on gametes, right? So you'll play with the words the way you'll understand this better, but the main keywords there with that 4 marks, the sperm cell is a gamete, you'll get that one. Or yeah, the sperm cell is a gamete, right? And then formed from meiosis. Byproduct, we are saying that it is formed after meiosis. So it's formed by meiosis. And then we know that after meiosis, a cell, it has to move from a diploid to a haploid, right? So with haploid, we know it's a half number of the total, which will be 30 divided by 2, which will be 15, right? So where is that 15 coming from? This is where the 15 you have to show, because they are asking you explain why the sperm of giraffe has 15, and we don't see 15 in our graph. So where now you also have to show us why are we asking this 15, right? So let's move on to 2.3.3. Yeah, rename the phase of meiosis where the halving of the chromosome number begins. Name the phase of meiosis. You know, right? We also have our PMET, right? You start with I, meiosis 1, then meiosis 2. We start with prophase, metaphase, anaphase, telophase, and we say prophase, metaphase, equator, anaphase, side, right? And then telophase, divide, right? So anything that happened before our chromosomes align at the equator here, metaphase, we have them at prophase. Then after the chromosomes start now to divide, right? In homologous pair, now they start to divide. So but name the phase of meiosis where the halving of the chromosomes number begins. So if we were saying that we have 30, show us where this 30 divide to 15, then we are going to say this will be anaphase, right? Anaphase what? Anaphase 1. Anaphase 1, right? So this will be anaphase, because if you just say anaphase, you are wrong. With meiosis, you know that you have to say meiosis 1 or meiosis 2, and if you say meiosis 2, you will be wrong, because during that time the chromosomes, they don't halved, the centromere do, right? The centromere, right? Those things between the chromosome. This is what they divide in meta. Yes, you did that term 1. Then from there, 2.4.3, describe the events in a phase named in question 2.3.3. Describe the events, right? What happened? 3 marks. What is going to happen? You are going to say the spindle fiber, and then you are going to say the spindle fiber is going to pull structures of chromosome to the different pool, right? Or a whole structure of a chromosome to the different pool, right? This is what is happening at the anaphase. Nothing happening here. So you will say it where now the spindle fiber will construct. You will say it, the spindle fiber will contract, or you say the spindle fiber shorten, right? Spindle fibers, right? Spindle fibers shorten, or we have the fancy name that they use. They can say contract, because you are also going to do this contract. We are also going to use this word during studying our eye, our human eye, right? There's an iris, or this thing that here, the black thing here. When that black thing comes together, we say it contract, and when it relax, we say it contracts. We're still going to learn that, right? So when you say something contract, it means that it's pulling, it's shortening, right? It's shortening. So the spindle fibers will shorten, and then they will say the chromosomes pairs. The chromosome pairs will be halved, right? Chromosome pairs will be halved, right? Moving to the opposite pole, right? Writing that, moving to the opposite pole, moving to the opposite poles, right? So if when now you say this, you are going to have your 3 marks, right? So I understand you know that. So this will be our final class here. This will be our final activity. Our time is up. We are left with 9 minutes. So here we also find a question number 3 last question paper, right? 2023, right? November 2023. You can see that we also have many questions that we have already covered that they asked on our final examination papers, right? So if we prepare thoroughly, I'm saying that we can come with this pass at the end of the year, because we also did this, right? A summer squash and what during the dihybrid crosses. If you miss our dihybrid crosses, go back and watch it, right? So another thing, we are here today. So here's the question. Right, with life science, you come and read the information that you are given, right? But the graph below shows the results of artificial selection for protein content in milli plants over 50 generations. So we have a table, and this table is showing the results of artificial selection. I told you that we have a natural selection and artificial selection. Artificial selection is the selection that is not happening naturally. You say that when you are good. And what is happening with artificial selection? I told you that this is an example where we go and take a pit bull and we mate it, or we interbreed. They say we interbreed, or we mate them. We force them to have sex, right? We have, this is underage. We force them, right, to mate. Then after mating, let's say maybe it's a pit bull and it's our sporty there. Those Lokshin dogs, mahayeng, the chats are mahayeng, right? Then we have a sporty pit bull, or we have pit bull sporty, or we have spore bull. Yeah, we'll have spore bull. Yeah, again we have pit bull. Then pit bull or sporty, we'll have spore bull, right? So that is an artificial selection, right? If it was naturally about what a pit bull was going to mate with the pit bull, right? If it was natural, but now it's artificial, it means that we, now as a human being, we are selecting to a group of individuals that we want to mate, right? And then this artificial selection here was done for protein content, right, in a millimole. So we have a millimole, and then this millimole we are crossing, or we are mixing 2 millimoles for a protein content, right, over 50 generations. So we come to the table. Where is the heading of the table? right? This table here, yeah, shorter, yeah, short. It has to have a table, it has to have a name, right? So the name of this, we are going to say this one, it will be a table, it will be a graph. Let's see from our previous, they say the number of chromosomes. So right now we are going to say the percentage of protein, right? The percentage of protein content, right? Protein content in a meal, content in a meal, in a mealy plant over 50 generation, right? Generations, you are done. Or we say protein content, it's just that the one that I was going to tell you, but I can't say it, right? Now they are being by hanahana, because back there we used to say protein, you come and say the word that they are using, the protein content, versus versus generation, protein content versus generation, right? It's a generation versus whatever we are doing this side, but that one I can't say it, right? So when now you are going to give a full heading. So let's go to our question and see if we can answer this in less than 5 minutes or so. 3.1.1, 3.1.1 but describe how this farmer did artificial selection of the mealy plant. Describe, just describe, or how this farmer did artificial. It means that this farmer mated, right? He mated or he interbreed. You say interbreed, it means that we are combining two gametes of two different, we are combining two plants, right? Or here we are talking about plants. So he interbreed or he mated two plants. He mated two plants that two plants that are having more protein content, right? More protein content, right? This is what the guy did, Velevel, right? He mated or he interbreeded mealy plants with a high protein content, right? At what time? Over 50 generation, right? So I'm trying to include all the information that we can put with this answer here, but describe how this, tell us how this farmer did artificial selection of the mealy plant. So this is what he did, right? He mated or he interbreeded two plants that have more protein content over 50 generation or over many generation, right? So number 1.1.2, what was the average percentage of the protein content in the mealy grains kernels at 15th generation? So here they want us to go at the 15th generation. Where's the 15th generation? You know, what was the average percentage? These are the average percentage protein content in mealy grains, right? Kernels, by the way, they are named kernels. So, but what was the average percentage of the protein content in the mealy grains at the 15th generation? Where's the 15th generation? We go here and check. As you can see, it's between 12 and 15. So the center here, it will be 12.5, 12.5. And then if you can see, it's not at the center, 10, it's at the center. So we can also go up. We can say 10, 5 here, 1, 2, 3, 4. Then we are going to say this, the second one, then it will be 12.8 there, right? 12.8 kernels. You say kernels. Kernels or percent, right? So they can also give you, maybe if you say 12.7 or 12.9, right? This is the range, right? You have to range here, right? So we can move on. Then 3.1.3, by how many times did the average percentage of the protein content in the mealy grains kernels increases between the 40 and the 50 generation? Show all calculation, right? By how many times did the average, this one I saw it on our memorandum here, I was checking it, and then here on our memorandum, I'm not sure, guys, I don't want to I don't want to find myself canceling or incorrecting the memorandum, because the memorandum is memorandum. But here's the memorandum. They're saying that it will be 20 divided by 14, and then we will get a 1.43 times, right? So let's go to our question. Then number 1.1.4, an artificial selection organism with desired characteristics are interbreeded and the genetic engineering. Okay, which one was this? This were describe one way in which 1.1.4 describe one way in which the process of artificial selection is different from genetic engineering, right? So you'll say that in artificial selection, we will interbreed. There's a word, a keyword, you interbreed two desired organism, and in genetic engineering, you manipulate or add or subtract gene, right, to produce a desired organism, right? There's a difference between these two, and then this is what they were explaining here. Where artificial selection organism with a desired characteristics are interbreed. This is the keyword, you interbreed in artificial selection. And then in genetic engineering, genes coding for a desired characteristics are inserted into an organism, or you manipulate the genes here, right? You are manipulating the genes. And then let's go back, our time is up. Thank you for being here, but I would like to leave you with this, right? I would like you to leave you with this at the comment section. Here they are saying that we have to find a percentage, right? But by how many times did the average percentage, by how many times did the average percent, no, we are not looking for a percentage, we are looking for how many times did this average percentage of a protein what increase, right? How many times did we find the increase between the year of 40 and 50 generation? Where's the year of 40? There's year of 40 and year of 50. So we want to find how many times did this average what increase, how many times, right? So we have how many times did this, you can say how many times did this increase from to 40. It means that they were going to 0, 5, 10, 15, 20, 25, 30, 35, 40, 45, 50. So in these 10 years here, how many increase? So if we can come and say 1, 2, 3, 4, 5, 6, 7, you can see that it increased 7 units, right? 7 units, or 6 if we start counting from here. 1, 2, 3, 4, 5, 6, right? I'm not sure, but they're saying that we have to come and say 20. Is that I can't explain to you why. Where is this? Why are we saying 20? This 20 here divided by this 14 here. That's what we are getting, and then the answer will be 1.43 times, right? 1.43 times. So they are saying that this increase here, it was 1.43 times. So we had 1.43, it will be 1 and a half mostly here, right? This one is close to, because we cannot use this 5, right? This one is close to half. So it means that there was a 1.5 increase times. So one time. So if maybe it was one time here and then this time here, so I'm not sure. I can't explain this 20 out of 14, guys. So let's go to our comment section and maybe try to explain this. This will be the question. This will be the question here, guys. Let's go to our comment section, hashtag 3.1.3, right? The question says, by, maybe I'm not getting the question, by how many times, right? 1. So they are saying one time, because if it's 1.43, just 1, right? That 1, that 4, you cannot add that times there to be 2. It's less than, right? You know what I'm talking about. 1.43, this one cannot add this one. If this was 5, we are going to, we say we are doing what here, right? The 1 is going to be that side, and then we are going to say 2 times. So it's 1.4, it's less than 1. So it's just 1 according to the memorandum. They are saying how many by 1. By how many times did the average percentage of the protein content in a milligrams camel increases between 40 and 20 and 50 generation? Show all your calculation. right? And then the marks there, it was only 2, right? So maybe this can also be the answer here. But this 20 out of 14, I can see that it's this 20 divided by this 14. Maybe that's how we divide in this graph. Anyone who have a better understanding, guys, show this to your teachers, show this to everyone who can help us, so that we can be able to explain why are we saying 20 divided by 14 and then have times. Whereas if we can say 20, if we can come and say 20 minus 14, we are going to have 6 units in between. So it means that it increased 6 times. It increased 1, and then after increasing, it increased again, after it increased again, after it increased again, after it increased again, after it increased again. Or we can be looking at this side, or it increased from here, it was increasing again, here it was increasing, and it was increasing, and it was increasing. But when we reach this 40, it increased 1 time, 2 time, maybe, right? But they're saying the average percentage protein content. So they want us to concentrate at this 20 and at this 14. Where are we getting it? We're getting at the 40 and 50. So with this increase, by how many times? Yeah, I think that one now. By how many times did this increase here? They're saying 1.3. So guys, thank you. That was it from me, guys. Don't forget to comment. Don't forget to like. Don't forget to subscribe, guys. Let's meet tomorrow. Tomorrow we'll be doing my mitochondrial links, right? So with that being said, and then tomorrow we'll be doing a revision, right? This term 2, so that guys come here, send messages, subscribe, and then share these videos to other learners, right? So thank you. That was it from me, Mr. Mpepu. Peace. Khutsong ayibelilina kurula kube nani. Thank you. Ukuthula. Peace.